Alright, so this model is going to describe mitosis, or basically the cell cycle where we divide cells. So in the cell cycle, before we begin the process of cell division, a cell is going to be in what we would call interphase. And in interphase we have a nucleus, we have chromatin, which is going to be loose on the inside of the nucleus. You have lots of other organelles, but basically in this first G1 phase, it's just normal living. It's growing, it's getting bigger, it's doing protein synthesis. You know, genes are getting turned on because this is nice and loose and able to be transcribed into messenger RNA, which then would get processed and then go and do translation at ribosomes. But at this point, we are not showing all of the chromatids. We are simply looking at a single chromosome. And this chromosome and this cell is diploid, which means that for this chromosome, there is a backup version of it. Di is 2, so they have a pair of 1 chromosomes. So these are going to have the same, or at least the same genes on each one. They might have different alleles of those genes or different versions of those genes, but that section is going to be coding for the same gene on each chromosome. So I know I'm using the word chromosome, but right now we're kind of thinking of this chromatin, which is the DNA wrapped around histones. And histones are the proteins that the DNA is wrapped on, and this is still fairly loose during G1. As we get to the S phase of interphase, during the S phase, we replicate the DNA and make sister chromatids. So we've talked about DNA replication. What we're doing now is taking all of that DNA and making a copy of it. And at this point, we now have 2 identical versions of this chromosome. So there's sister chromatids here, and there's sister chromatids here. So we went from diploid, and again it's still diploid, but we have now twice as much genetic material here because the DNA has been replicated. And the whole process of mitosis, which we're about to get into here, is to take it from diploid and finish the exact same way we ended. So mitosis is happening in all body cells, and after that initial fertilization, we replicate, we go through mitosis to go from one fertilized cell to trillions of cells within the human body. So, and again, humans are going to have 23 pairs and not just 1 pair, but it makes it a little bit simpler not having all of the chromosomes in here. So we're going to begin mitosis now. And with mitosis, the first stage is prophase. And sometimes it's broken up into prometaphase and all of that kind of stuff, but basically what's going to happen is we are going to take these, which are a little bit more like spaghetti, and turn them more into sausages. So we're going to condense the genetic material and package it tighter so that it is much easier to pull apart. So I'm just condensing the chromosomes. And this point, when they become coiled up tightly, now we're actually, the word chromosome is the proper word. And again, this is still a chromosome, but it's attached to its sister chromatid. That's condensed, and I'm going to condense this one as well. Alright, so again, we have sister chromatids, but these are what we would call homologous chromosomes. And another part of prophase, after we form these sausage tightly condensed chromosome structures of DNA, we're going to lose the nucleus. So the nuclear membrane is going to dissolve, and now this is more free floating. Another part of prophase is our centrioles are going to migrate to opposite ends of the cell. And they are going to start sending out spindle fibers. So they're going to send out these long microtubules, protein filaments, to ultimately get into cell division. So at this point we are basically done with prophase. The chromosomes condense, the nuclear membrane dissolves, the centrioles migrate, and they start sending out spindle fibers. When we say we're in metaphase, are when the sister chromatids line up, all of these chromosomes line up at the metaphase plate, or the middle. Meta means middle, so they're lining up in the middle. And again, we need these sent out from either side, because ultimately we are going to pull apart the sister chromatids. So at this point, this is another really important checkpoint to avoid non-disjunction. Non-disjunction would happen is if these things don't pull apart and divide evenly. If these would both go towards this side would not have the full amount of genetic material, and this side would have too much. So a very important checkpoint in cell division right here is this metaphase checkpoint. We have a little tug of war going on here. Before we separate the sister chromatids, we got to make sure that spindle fibers are attached correctly. This would be metaphase when they're all lined up in the middle. At anaphase, they pull apart, and the genetic material moves towards each side. Ana is apart. I'm pulling these guys apart. And now tele is 2. So during telophase, the nuclear membranes come back and envelop the genetic material. Again, we started with one pair of homologous chromosomes. So now in telophase, it has basically 2 nuclei within one cell. And that's basically what we would call the end of mitosis. Cytokinesis is when we actually split these things apart. And we pinch this together. We pinch the outer cell membrane together, and now we've just divided the cell. We've made exact twin copies, and we finished with the exact same genetic material, identical genetic material in the beginning in the end that we began with. We just have 2 new cells now. And again, you see once we split it, now if we go back into the cell cycle, after cytokinesis we go back into the G1 phase, where things would grow. So then you can see once we split this how much smaller it gets. So if we continue to just do mitosis without a G phase, we'd get smaller and smaller and smaller and smaller and smaller. But what we want to do is when we get done with mitosis or cytokinesis, these need to grow larger again, and then we have to duplicate the DNA. We got to check for mistakes. It needs to grow a little bit more before we end up condensing things back into chromosomes and getting rid of our nuclear membranes and get into mitosis again. But again, mitosis, make it twin, 1 division, and identical material that we finish with that we have began with. And that's mitosis.